Thank you everyone for attending, and thank you for the organizers today to offer the possibility to present the work we conducted since about 10 years at Toulouse in France. I decided to change a little bit the subject of my talk to present the main research effort that was driven in France that is called Sunrise, as sunflower resources to improve yield stability in a changing environment, that I coordinated from 2012 to 2020. And I thought it was the opportunity at this International Sunflower Conference to present you a little bit of this project, and also then having a second part to present you an example of what we can do to better understand the genetic basis of drought tolerance. Okay, of course, a little bit of context with the climate change impact on sunflower. We've seen a very nice presentation this morning with Philippe Debac. There are different predictions on how climate change would impact sunflower production. What is original here compared to what Philippe presented earlier this morning is some data that we collected during our project, in the Sunrise project, where we were able to estimate the impact of 1 day of stress on sunflower. And when we make the figures, and here the figures can be discussed, but at least they are estimated using crop modeling. Given the acreage of sunflower in France, 1 day of drought in France is really costly, up to 30 million euros for farmers. So this is a major factor limiting sunflower production and having a major economic impact. And we were also able to estimate using central crop modeling that in our network of experiments we modeled 35 days of drought stress. And in the 30s, in 15 years from now, 10 years from now, the increase would be up to 50%, up to 50 days of drought in this network in France. So the impact is major. So this project, this Sunrise project, was constructed as a partnership between breeding companies, Technical Institute Terre-Novia and INRAE and university labs. So there were 16 partners, and they are mainly concentrated in the southwest of France, which is an important region, production region for sunflower in France, with different expertise. And what is very important is the multidisciplinary approach that we gathered and that Philippe explained already this morning. So the scientific goals of the project were really to study drought tolerance, developing the tools and infrastructure to be able, on the long term, to study this impact at the physiological and molecular levels, and also in the context of heterosis, because of course sunflower is an hybrid crop, and hybrids respond differently to environment than their parents, and it is very difficult to predict the behavior of an hybrid from the parental values. So in this context, we had three main scientific axes. One is an approach using genomics and systems biology to understand how sunflower responds to drought. A second approach was combining genetics and crop modeling to really study the genetic basis of tolerance, but in agronomically realistic conditions, and being able to predict and estimate the effect of the genetic innovation that we found in using these crop models. And a third approach was a socioeconomic approach to understand really different aspects of the biotechnology of sunflower. So just quickly on this approach, we had results by estimating the ecoservice pollination provided by sunflower, the economic impact on farm incomes. We also studied on the economic level the valorization of the oil cake in sustainable development, and also conceptualized eco-innovation in the plant breeding domain. And these legal issues were and the economics were studied in France, but also at the European level, for instance in Hungary and Romania. On the impact of sunflower results at the genomics and systems biology level, we had a range of major results. First, at the beginning of the project, Stéphane Munoz developed different genotyping chips using genomic resources that could be incorporated by private partners. We developed, they allowed the description of genetic resources, and I've already presented yesterday some example of this. And of course, this led to new research projects on Orbanki and genetic resources. In 2016 and 2017, we produced the first high-quality genome sequence for sunflower. And this had, of course, major impacts. We identified in this article that I invite you to read the genomic regions involved in the breeding history of sunflower. They having a reference, really accelerated breeding and positioning of molecular markers on the genome. And I invite you to go into the genomic web portals, and especially including the Allele Atlas that was released recently, and the Cell Express portal that also was released very recently, and that gathered all information, structural information and structural diversity at the genomic level, and all transcriptomic data ever published in sunflower. And of course, the genome, we sequenced it using a very innovative approach, using PacBio sequencing. And this led to a patent application. This led also to sequencing other genomes in collaboration with the International Consortium for Sunflower Genomics. And so now we have 8 cultivated lines with high-quality genomes and several wild sunflowers. And of course, other plant genomes were sequenced using this methodology, such as the Orbanki genome. Okay. In this paper, we identify pathways controlling oil content. We identify the different events of genome duplication and the paleogenomics of sunflower, and there is a section on the genetic control of flowering time in sunflower. This led to another article in collaboration with Canadian and US scientists on the pangenome for sunflower and Helianthus. Okay, so there was this other axis, scientific axis, integrating genetics and crop modeling. And on this scientific approach, we developed on one hand phenotyping tools. So the Eliafin platform, we developed drone phenotyping and the phenomobile phenotyping in collaboration with the Phenome project. And we did stress modeling to describe genetic by environment interaction, and I will present an example later. And we identified genetic control of tolerance and provide new genetic material, including introgression lines, more tolerant to drought. Sorry about that. And as I mentioned earlier, we did genomic prediction of flowering time, but also of drought tolerance in the genetic resources. So we had different genetic designs in this project, and 2 were original: an incomplete factorial hybrid panel that was an incomplete cross between 36 sterile maintainer lines and 36 restorer lines. All were resequenced, and we could impute the genotype of each hybrid based on the resequencing of the parents. And a nested association mapping population from 9 parents across 4 times each, and that led to 576 recombinant inbred lines that were crossed to 3 testers. And this was genotype using Axiom chip developed in the frame of the project. We also developed an allelic diversity platform based on introgression lines from wild Elianthus anus introgressed into the reference cultivated line XRQ. And here was the choice of the different introgression lines from the 4 different genetic groups identified here. And this led to marker-assisted selection of the different lines. So we ended with about 500 introgress lines, each genotyped and carrying, as you can see here, all the different parts of the genome in the different lines. So now I wanted to present you an example on how we can use all these resources and infrastructure to better understand the genetic control of drought stress plasticity. So I invite really you to read this article from Brigitte Mangin, published in 2017, where we describe the philosophy of our approach. So in this article, we use the core collection representing the genetic diversity. So here of sunflowers. So here in red, the core collection, and in blue, you can see the genetic resource center diversity that we have at Langlade. So this core collection represents a large part of the genetic diversity present in cultivated sunflower. This was a genotype using an Axiom chip, and phenotype in collaboration with our partners, breeding companies, in 17 environments in France. And we used the Sunflow crop model to estimate the different drought, cold, and nutritive stress scenarios in all these 17 environments. Then we were able to work out, to calculate response for each line to the different stresses. And this slope and this response to the stresses is what we call plasticity. And so we add the yield plasticity to drought, cold, or nutritive stress. This GWAS genetic genome-wide association study allowed us to identify 9 regions that are associated to yield tolerance to cold, 6 to drought, and 21 to nutritive stress. In this article, we present the results of the 9 genomic regions for cold, and we are still working with our partners on the remaining regions. In the Sunrise project, we had a similar approach on the different genetic designs that I presented, the incomplete factorial design that was resequenced and phenotyped in 13 environments. This allowed us to identify 3 QTLs for yield plasticity, 7 for drought, and 9 for nutritive stress. Similarly, for the nested association mapping population, we did a similar approach. We identified 33 regions associated to cold, 17 to drought, and 18 to nutritive stress. These different regions, we don't find always the same regions and the same number because the trial networks are different, and sometimes we had more cold stresses, sometimes more drought, etc. And we had a last genetic design, which is a kind of classical one, a biparental recombinant inbred line population that was phenotyped, but this time we didn't phenotype it on field trials, but on the Eliafen high-throughput phenotyping platforms, carefully controlling drought scenarios from flowering to the end of the maturity of the plant. And this led us to specifically identify QTLs involved into drought tolerance, and here we identified 4, and this was published in 2019 in the article from Florie Gosseau. So we are now having all these different regions, and when we really want to know if we find the same regions, if we want to integrate all this data, we were able to identify 34 QTLs that we estimate are validated, because we can find these regions are involved into yield plasticity in at least 2 genetic design and 2 completely independent genetic design and trial networks. Some of them were also found in 3 different genetic designs. And so here you have 7 regions involved, found in 3 genetic designs, and the rest is in at least 2. Among them, there are 2 regions. One, the deep 3 region, which is drought yield plasticity chromosome 3 region, that are already presented at several occasions, and specifically the Symposium on Sunflower and Climate Change introduced in 2018, and the deep 12 region that was already described in the Gosseau 2019 article. So I will we'll take this example of the deep 1 region to show you a bit what we can do now. Thanks to all this data, we can colocalize different SNPs that are associated to different traits in the region. So here is an example on this region. So in blue, you have the original SNP identified in the Gosseau article. And you can see 4 genomes, high-quality sequenced genomes that are reconstructed and that are available on the web portal on the top of the slide. You can see that some genotypes carry a large deletion, such as the PSC8 genotype, and all the small blue lines here correspond to genes that are annotated, and you can see there is actually quite a diversity of genes in the region. And so really this is key to have these high quality genomes for the different genotypes that we work with, to understand a little bit, to understand really how, which gene is important and how they work. So we developed near isogenic lines to characterize better this region and its impact on drought tolerance. And here I would like really to emphasize the role of Nicolas, who's responsible for the high-throughput phenotyping platform ELIAFN, that you see the small movie here that describes a little bit what it does. And you see on the right the seed weight, so the yield of plants, with either the low plasticity allele on the left and the high plasticity allele on the right, and in irrigated or stress conditions. So here irrigated is in red and stress in blue. And you can see that according to the allele on the locus, the response is completely different. So we can, using this, really validate these results, although we had different, the effect from the previous data was validated in different trials, but here on the platform we can reproduce the effect of the allele, and we can look for new alleles in the introgression lines that we developed. So here, for instance, we had the 5 populations, and we identified using the genotyping using Axiom which line carries the wild allele. And you can see that on the plant weight, some introgression lines respond very differently. Some are plastic, some are stable. And on the final seed weight on the bottom, you can see a diversity of yields in the different introgression lines. And so now we are replicating these results to have several years of experiments on the ELIAFN platform. This also confirms that Deep 12 is acting on the biomass reallocation to seeds in drought conditions. If we want to go further on really which gene in this QTL is controlling drought tolerance, we can study specifically the gene expression of all the genes here in the region. And here is a few examples of how some genes of the region respond to drought according to the allele. So it's the same thing than earlier. On the left you have the low plasticity, on the right the high plasticity, and you have control and drought conditions. And you can see that the genes respond differently according to the allele to drought. We collaborated with a statistician from Israel. Okay, and so we were able to infer a gene regulatory network of the genes part of that are included in this region, but that respond in general to drought, and that include also other QTLs. And so we could include them into this gene regulatory network. So this integration and understanding really how all these genes and QTL work together is really the next challenge for us, because here when we come back to all the results we have, the results I showed you, we are only starting to scratch the top of the results, because we all know that drought response and drought tolerance is a complex and multigenic response. So you see that there is still a lot to do to understand which gene and how they work together provide drought tolerance in sunflower. So a little word on the perspectives that I can foresee now. As you understood, the project is finished now, so now we really, I think the challenge is to identify the actual genomic polymorphism that are behind these QTLs, whether they are structural polymorphism or more SNP and polymorphism. Thanks to the integration lines and different methods, we can identify the physiological processes that are involved in drought tolerance. And here we have an example on the harvest index and the carbon reallocation to seeds during the drought stress. And I think an important step now is to validate a new cropping management system that imply new stress scenarios, how this genetic control really works, and if it can be transferred to this new cropping system, because everything I presented here was done in conventional cropping system, and now with a more agroecological cropping system, it is important to validate this in this case. And for this, we really I want to stress that we need interdisciplinary approaches, and this is what I wanted to exemplify today, is the complementarity between quantitative genetics and agronomy, crop modeling, and genomics and systems biology. It's really this capacity to go from one high level in agronomy to molecular level in genomics that will bring innovation that we need for sunflower tomorrow. And so for this we need to describe better the structural variation existing in sunflower, to improve our high throughput phenotyping capacities and related to ecophysiological processes, and included it in agronomy and crop modeling using the same variables when we are doing ecophysiology and molecular biology and agronomy. And here, all this was possible only thanks to a public-private partnership that was very important, and that I think should go on. This allows our result to be closer to the production in the in farms for farmers, and also to the stress scenarios that are really happening in the farms, and not to be too disconnected from the production. And because of this strong public-private partnership, this can accelerate breeding. Otherwise, it is difficult to make the links between the communities. So, as you guess, there is really a lot of people to acknowledge. So here I really want to thank three persons. Pierre Casadebaig in the crop modeling, because this is really, as you understood, all the importance of describing the stress scenarios. Brigitte Mangin, who's a quantitative geneticist and statistician, that retired now, and that really was a key in all the Sunrise project, and Florie Gosseau, who's a PhD student who really integrated all these disciplines in relation to the results I showed today. I want really to thank Stéphane Munoz for all his help during these years in genomics, and the partners, the breeding companies that are listed here, and the funding agency, of course. And thank you for your attention.
Thank you, Nicolas.